Dziękuję bardzo za miłe zaproszenie. Też chciałem podziękować ambasadzie i profesorowi Żygockiemu za sponsorowanie dzisiejszego lokalu. Mam około 20 minut, muszę się trzymać do czasu, więc będę się starał tak zrobić. Wiem, że jest dość zróżnicowane towarzystwo, więc też muszę trzymać to troszeczkę prościej i nie za bardzo się wgłębiać tutaj. Więc zaraz się przerzucę na angielski. So the area really is metabolism, cancer metabolism, and how cancer cells grow by using fuels, and specifically a fuel called arginine, which you're probably very familiar, is one of the 20 amino acids that are used to build proteins, and it actually turns out to be a non-essential amino acid for normal physiology, but for some cancer cells, they require it as an essential amino acid, so this is the trick to try and exploit that in therapy. So this may be familiar to some of you, but it's essentially a sort of roadmap or atlas of various features of cancer cells. And I've sort of imposed on there some of the recent, maybe not so recent developments in our understanding of cancer, and you can see the Nobel Prize winners featuring there. I've specifically chosen Otto Warburg, who is a German researcher, physician, who first described fermentation of glucose under plentiful oxygen, so a very unusual situation of aerobic glycolysis, because normally you get glycolysis under low oxygen levels. And he got the Nobel Prize in 1931, and if you recall the other week, there was another Nobel Prize on oxygen sensing. Three researchers shared that Nobel Prize. So clearly a very topical area of metabolism, and really that was one of the first characteristics of cancer that was developed. But going back, actually, Maria Skłodowska-Curie first described radium and polonium. Of course, radiation can trigger cancer, but is also used in cancer therapy. And I'll come back towards the end of my talk on the Nobel Prize on immunotherapy, because that also ties into some of the work that we're doing at researching this area of arginine metabolism. So Jim Allison and Tosuku Honjo there. So, what's the difference fundamentally in metabolism of cancer cells compared to normal cells? Well, normal cells will use most of the sugar, for example, or glucose that we eat, to produce ATP, which is obviously used for energy. So without ATP, we can't move, we can't talk, etc. And you can see there's a much larger amount of that glucose is used for energy production. But in the cancer situation, cancer cells actually sort of almost a 50-50 split. So yes, they do produce energy, but a lot of that carbon in the glucose is being used to produce mass. I mean, that's how cancer kills. It's by proliferating. I have a patient at the moment on the ward who weighed 85 kilos. He's now 100 kilos, and at least 15 kilos of that is his melanoma. It's just expanded so much. And that's because cancer cells have a voracious appetite, and here they're using glucose. So we're interested in another fuel, arginine. So unlike glucose, arginine has a much more diverse role in various metabolic pathways. So of course it's used to produce proteins because it's an amino acid. It also is used to make nitric oxide. It's used to make other various molecules such as polyamines, used to make other, as a precursor for other amino acids. And also, one of the advantages of why these tumor cells stop making arginine and require it as an external fuel is because the precursor aspartate then gets shunted into producing nucleotides. So this is basically what we think one of the roles for switching off a cancer cell making arginine is for this proliferation pathway. So how can you exploit this? Well, if you have a cancer cell that feeds on arginine from the bloodstream because it doesn't have the enzyme that makes arginine, you can starve it by using various drugs. And the drug that we work with from a company in California, Polaris Pharmaceuticals, they make this enzyme which can convert arginine into citrulline. And our normal cells can reuse that citrulline to make arginine again, but the cancer cells can't do that. So that you're starving them and they undergo cell death. And there are some cancers, particularly aggressive ones, as listed here: melanoma, mesothelioma, which I'll come on to, ovarian, platinum-refractory ovarian cancer, pancreatic cancer, and so on. They have very low levels of the enzyme that makes arginine, so these are tumours that you may think you could starve by removing arginine. So one of my main clinical and research interests is mesothelioma. Unfortunately, in the UK, we have the highest death rate of this disease. A lot of asbestos, it's an industrial disease, a lot of asbestos has been used in buildings, including hospitals and schools. And you can see it grows and basically constricts the healthy lung, and then later on can spread. And you can see there's a very modest improvement of 2 to 3 months survival from 9 to 12 months with current chemotherapy. And that really is still the only approved treatment for mesothelioma, although some immunotherapy drugs are beginning to emerge in this area. But essentially, this is still the only treatment approved in the NHS and worldwide for mesothelioma therapy. So, if you're going to impact on that, you'll be able to hopefully shift that curve to the right. And these are the various asbestos fibers that are used in inducing mesothelioma. So there are different types of mesothelioma. Some mesotheliomas are more aggressive than others. The most aggressive is actually this type called sarcomatoid mesothelioma, which has a spindle cell appearance down the microscope. And if you're unfortunate to develop that mesothelioma, your average survival is 3.5 months, median survival is 4 months. So a very aggressive cancer, and some places would not actually even treat patients with sarcoma, so it's saying, well, we'll probably just send you to the hospice. So we studied mesothelioma some years ago, and actually this was towards the end of my PhD, but we found that half the mesothelioma cell lines lacked the enzyme that makes arginine. And if you removed arginine, here we're doing that with the drug ADI-PIG20 from Polaris, you can see they're very sensitive, and by 6 or 7 days they're mostly all dead in the Petri dish. And you can select a patient on their biopsy, do a protein test, and see if it's a tumour that doesn't have the enzyme or has the enzyme. And these are the patients we then selected for a clinical trial. You can see this was confined to 8 places in England. It actually nearly killed me doing this clinical trial. But anyway, I won't go into all the details of that. This was almost just over 10 years from when we were awarded the grant to when we actually published the paper on this study. So it was around 70 patients were randomized 2 to 1 to either receive the drug that removes arginine, it's a weekly injection into the muscle, or patients were followed up with best supportive care, so they didn't receive the drug. And we compared the 2 groups and scanned those patients to see simply were we reducing the growth of the cancer by removing arginine, this fuel, from the bloodstream. So, pharmacological rather than dietary approach. I mean, dietary would be very difficult to remove arginine from the body, but this enzyme, as you saw in the Petri dish, really rapidly reduces arginine. So, we did this study, and the drug does what it says on the tin. It rapidly downregulates arginine, but you can see over time the arginine level starts to come back in the blood. So it's really only, you have this window of 4 to 8 weeks, and the citrulline, as you would expect, goes up because it's the degradation product of arginine. And you get no change in the patients who are just being monitored, who don't get the drug. The arginine citrulline levels essentially stay the same. And the reason for the arginine levels coming back up is because the drug is a foreign protein made from bacteria, and it produces a neutralizing antibody response. So that's a pharmacological issue relating to the drug. But we could see on a PET scan where we monitor a radioactive glucose molecule. This is before treatment with the drug that starves arginine, and after that we get some impact with the tumour using less glucose in terms of this particular analysis. So the main endpoint for this study was reached. We had a modest improvement in progression-free survival, so that simply means that as we were monitoring on the scan, patients who received the drug had 1.2 months extra disease control compared to those patients who had no specific treatment for their cancer. Within 2 months, the average scan at that point had already shown progression. And we looked at overall survival. The study was actually not powered statistically to show that, but there was a trend towards an improved survival in those patients who received the drug compared to best supportive care. So we wanted to try and optimize this treatment, because clearly 1.2 months of progression-free survival, is statistically significant, but is it clinically meaningful? And we studied whether we could enhance this effect in the lab, and essentially this slide summarizes about 5 years' work, £1 million, which was a Cancer Research UK fellowship. And what we showed in this work was that by removing arginine, we could improve the activity of one of the main drugs that's used for mesothelioma, which I mentioned at the beginning, the 2 chemotherapy drugs, pemetrexed, which is an antifolate. And you can see pemetrexed has almost no activity in this cancer cell line model. Arginine starvation with the drug is actually very effective out to 12 days. The tumours in the mice are actually reducing. And there's an additive effect by adding the 2 drugs. So that's what we explored in the next clinical trial, was basically combining the standard treatment for mesothelioma and also lung cancer at the time, pemetrexed and cisplatin, with the 3rd drug, ADI-PEG20. What we noticed in this study was that by removing, by adding in the chemo with the arginine deprivation, we actually got much more sustained starvation of arginine in the bloodstream. The citrulline levels also were maintained out to 18 weeks, which is how far we sampled these patients. And you can see the neutralizing titer of antibodies did increase, but it plateaued much earlier on. And what was striking about this small study of 9 patients was that we had a 100% disease control rate. And you can see that 4 of the 5 mesothelioma patients, I mean, caveat, this is a very small study, but 4 of the 5 patients had the more aggressive, 2 of them had the sarcomatoid, which is, as I say, median survival 4 months. And the overall survival median in this small study of 9 patients was 14 months. And you can see a patient here with sarcomatoid mesothelioma who, in some parts of the country, would have been just sent to the hospice, had a marked reduction in the tumour. You can see this rind really shrinking. And he suddenly started to see, to be hopeful for his outcome, and actually bought himself a car. He was so well, but unfortunately, 7-8 months in on the study, he developed 2 brain metastases. So it shows you how aggressive this tumour is. But he had initially a very good palliative response to the treatment. So based on that, there's currently a global trial running, of which I'm the chief investigator, called Atomic Meso. That's actually an acronym I developed while I was skiing this run in Canada about 20 times, and suddenly I thought, Atomic, this sounds a great name for a trial. And we've just about reached the first 176 patients. It's a global study being done in the UK, big centres in the US, in Australia, Italy and Taiwan. And this is one patient who sadly just died this month, but her goal was, she was told she had less than 6 months to live, and she made it to 2 years, and her main goal in life was to see her daughter graduate with her master's, which took place in May of this year. So Fiona Holmes, keep patient and may she rest in peace, but she had a very good benefit on the trial. Of course, we don't know if she had placebo or the drug. This is a blinded study, so neither the patient nor the doctor knows who's on what treatment. So in the last sort of 5 minutes or so, I just wanted to go through what we want to understand is the resistance to arginine starvation, because clearly there is an impact. It can be improved, the current chemotherapy can be improved by combining arginine starvation, but what are the resistance mechanisms? If we could understand the resistance that develops, we could optimize the treatment further. And one of the main mechanisms of resistance, of course, is if the tumour doesn't have the enzyme to make arginine, well, it will eventually switch the enzyme back on. And we've seen that in the tissue culture, in the Petri dish. If you give the cancer cells the drug, over time they will re-express the enzyme ASS1, and this is actually due to an epigenetic modification, so demethylation of the promoter. And actually, in some of the biopsies that we've taken from patients, we also see, here's probably the most striking example, a patient with sarcomatoid, very negative for the enzyme. You can see that after, I think it was taken around 5 or 6 months, you can see a lot of re-expression of the enzyme that makes ASS1. So one strategy would be, could you prevent this enzyme from being re-expressed? The second sort of pathway, actually a Nobel Prize was given for this to a Japanese researcher a few years ago, so autophagy. Autophagy is essentially a way of cells under stress undergoing sort of breakdown of organelles to replace their building blocks and so the cells can survive under starvation by, for a limited period of time by breaking down their own organelles to resupply nutrients. And we found, and several other groups have found, that by adding chloroquine or anti-malarials, this sort of detergent type of effect of these drugs can prevent autophagy, and you can get improved control. So this is something we've done in lymphoma cell lines. This is some work in mesothelioma cell lines by an Italian group by adding in also chloroquine. And here's a sarcoma model. You can see very aggressive, but there is some benefit with arginine starvation and a modest improvement further if you combine it with chloroquine. But you can see the tumours are still growing out in this particular, so it's even more aggressive than the previous model I showed you in mice. So in this study, we think we had a signal of this effect because this is a study we recently published at the beginning of this year in patients with relapsed high-grade brain tumours. So you might be aware, astrocytomas grade 3 and glioblastoma multiforme. So these were patients who had already received standard of care. Median survival at that point is usually around 6 to 8 months. And actually, what was characteristic of this small study of 10 patients, that these patients had all, essentially, apart from 1 or 2, had already had second and even third line chemotherapy. So this would be a group where you'd expect the median survival to be more around 4 months, probably. And we had a median outcome of around 6 to 7 months, which is what you'd expect in second line. In fact, 2 patients did very well, and one patient here who was still alive at 24 months, has also recently died, but he was, this month would have been 36 months, so he would have been 3 years, so he'd have died around 34 months from starting on the trial. And he was very interesting. He was so well, he flew twice over a period of a year to visit his family in South Africa, but he had, you can see this frontal glioblastoma, de novo multiforme tumour, which started to grow after a while he was receiving the arginine starvation and the chemotherapy. And if he'd hit 20%, he would have come off study. And around this time, he was complaining of leg cramps. And I'm sure if there are GPs in the audience who prescribe, you don't think twice, you almost write out the quinine sulfate, which is the standard treatment for leg cramps. And we noticed the tumour then to start shrinking. So at this point, he was just on the arginine starvation drug and quinine sulfate, actually reduced to less than 5% from his original baseline tumour, but then started to regrow. And at that point, when it hit 22%, we had to take him off the study. So, there's quite a bit of interest in looking at blocking autophagy with these drugs. And you can see his tumour, in contrast to the patient with mesothelioma that I showed you on rebiopsy of his brain tumour after he came off the study, there was still no expression of the enzyme. These areas of staining of the enzyme are actually blood vessels, so they're an internal control, and our blood vessels normally express abundant amounts of arginosubstrate synthetase, because as I said, that's involved in nitric oxide production, and nitric oxide is a key molecule for maintaining vascular stability. So certainly re-expression was not the cause of his progression, but autophagy possibly was involved in this situation. The third mechanism which we're particularly interested in the lab is how tumour cells can co-opt or attract other so-called normal cells, so macrophages would be a particular one, or fibroblasts, and these could act as a supporting cell. So here we have a cancer cell being deprived of arginine. It sends out chemicals to say that it's under stress. Please feed me. and it attracts sort of traitor cells, if you like, or macrophages in this situation, and they're actually providing the substrate, maybe arginine or argininosuccinate in this situation, bypassing the effect of this drug. So we've got a paper at the moment in preparation, hope to get that accepted soon. But this also leads into the other side of the immune system, so that the macrophages are part of the so-called innate immune system, but here the adaptive or antigen-specific part of the immune system, it's quite controversial whether removing arginine is a good or bad thing. So in this study from Switzerland, for example, very recent, published from 2016, I think that was, and they're saying that arginine actually can modulate T-cell metabolism and can improve anti-cancer therapy that's based on the immune system. A paper from a collaboration group in Israel, Eris's group, have suggested that tumours that have problems with ASS1 or so-called urea cycle defects may actually have more efficacy with immunotherapy. So we have looked at this in the lab, and also the company Polaris published a short paper recently suggesting that arginine deprivation can improve some of the signals on cancer cells that may allow some of these immunotherapy drugs to work, particularly PD-L1, a molecule on the surface of the cancer cell. And we have data, which I'm not showing here today, but essentially in an aggressive lung cancer model. This is from the publication in a colorectal cell line that was negative for the enzyme arginine. You can see that by combining arginine deprivation with those immunotherapy antibodies against PD-L1, you had better control in the mice. And based on work that we've also done and also shared with Polaris, we're starting a new study in the new year, which will be just in patients with lung cancer called ITRAP, and that will be combining the 3 drugs that I've already mentioned to you with one of these checkpoint immunotherapy drugs that the Nobel Prize was given for medicine last year in immunotherapy, so atezolizumab, which is made by Genentech. So this is an academic study, but drug provided by Polaris and Genentech. So in summary, I hope I've shown you some data that convinces you that mesothelioma that has low levels of the enzyme that makes arginine ASS1 is a good target for arginine starvation. We've shown in a larger study that I haven't really gone into today, of 30 patients, that the aggressive subtype of mesothelioma seems to be particularly sensitive for arginine deprivation, and that's the current population of patients in the atomic study. And we believe that understanding resistance will be critical to improving arginine deprivation, and hopefully that will become a new approach in the clinic. Just as you may be familiar, asparaginase was introduced in the 1960s for the treatment of childhood acute lymphoblastic leukemia, and that's now a standard therapy for children with ALL, and survival has gone from 5% at 5 years to over 90%. And again, that's a drug that's used often with 3 or 4 other chemotherapy drugs. So I'd just like to acknowledge all the people over the years in the lab, also my PhD supervisor, Fran Volkwill, Nicholas Moyne, who's the head of the Barts Cancer Institute, past and present members, the various the families and patients involved in all the studies, the medical teams, and also Polaris Pharma, particularly Dr. Golmaski and Dr. Ball and Wu, for all the support over the years, because without the industrial collaboration, we would never have got this far. Obviously, all the funders that are listed there. Thank you very much for your attention.